Fellow of International Society of Biotechnology, Indian Society of Mycology and Plant Pathology. He has published more than hundreds of papers. He has put in more than 25 years of teaching and also research. And he has guided quite a good number of PhD candidates. He has authored so many book chapters and he is also being honored with Young Scientist Award. He is also an editorial board member of many reputed journals of international repute. Dear students, a senior professor with such a high profile has agreed to address you, and we are really happy and proud to have Professor like Umesh with us, who is here to address our students. I extend a very warm welcome to you, sir. Umesh sir, I welcome you again. Next, I would like to welcome Professor B.M. Subraya, Honorary President, Sarada Vilas Education Institutions, and Sri N. Chandrasekhar, Honorary Secretary, Sarada Vilas Education Institutions, Mysore, for their encouragement in hosting such webinars from our college. Lastly, I would like to welcome each one of the participants for joining us in this webinar. I wish all the success for this webinar, and I request my dear students and also the students who have participated from other colleges to make use of the expertise of the speaker, interact with the speaker, and make this webinar fruitful. I thank all those who have worked directly or indirectly in organizing this webinar. Thank you, one and all. I wish a great success. Thank you again.
Thank you, ma'am, for your warm address. Now, I call upon Kanika Gupta to introduce our today's guest, Dr. Umesh.
Thank you. Dr. S. Umesha, serving as a professor in Biotechnology, University of Mysore, and earned his MSc, MPhil, PhD from the University of Mysore. He has been an elected Fellow of prestigious National Academy of Biological Science, Fellow of International Society of Biotechnology, Indian Society of Mycology and Plant Pathology. He has undergone his postdoctoral training in the Denmark 2002 under prestigious Danida Fellowship and Virginia Tech University, Virginia, USA, and a prestigious DBT Crest Fellowship 2007-8. He has also been invited to present his research finding at the international conference in Germany, Malaysia, and China. From the year 1999, he is serving in the Department of Biotechnology and continued his teaching in the research in the area of microbiology, food and environmental biotechnology, and plant biotechnology. His research interests include plant biotechnology, molecular diagnostics of plant, animal, foodborne pathogens, host pathogen interaction, cell and tissue culture technology. He is the coordinator of the DBT supported MSc Biotechnology program of University of Mysore. He has received several competitive research grants from different funding agencies such as DST, DBT, UGC, ICMR, and East West International Seeds Private Limited, and attracted many young PhD candidates. 25 MPhils and 12 PhD candidates have already awarded their degrees under his supervision and adjudicated 50 PhD theses of other universities, including IIT Delhi. He has published over 130 research papers in the peer-reviewed journals of international repute, including Scientific Reports Nature publication. His H index is 25, I-10 index 47, with over 2040 citation, and he has presented more than 80 research papers in the national international conference and also chaired many scientific sessions in the conference. He has authored one book, Plant Biotechnology, 5 book chapters, and organized one national, one international conference, along with one refresher course in UGC HRDC, University of Mysore, and has been in the editorial board and reviewer of many scientific journals of national and international repute. He is the recipient of the Professor K.S. Jagdish Chandra Young Scientist Award from the University of Mysore 2001, Professor V.P. Bade Memorial Award from ISMPP Udaipur, and is the life member of the Indian Phytopathological Society AMI, ISST, and Indian Science Congress. Thank you for giving me such a wonderful opportunity to speak about a great personality, Dr. S. Umesha. Thank you.
Thank you, Kanika, for your brief introduction. Now, I call upon the speaker, Dr. Umesh, Department of Biotechnology, Manasa Gangotri, to take over today's session. Over to you, sir.
ಎಲ್ರಿಗೂ ನಮಸ್ಕಾರ. ಗುಡ್ ಆಫ್ಟರ್ನೂನ್ ಟು ಒನ್ ಅಂಡ್ ಆಲ್. Am I audible, madam? Respected principal of this great institution, Professor Devika madam, and all other staff members, and the president of the society, secretary, and other EC members of the society. Most importantly, my beloved students. First of all, I would like to take this opportunity to thank profusely Professor Devika madam and all other staff members for having invited me to discuss some aspects about biotechnology to the young students, young minds. If you get to know some of the information, it will be useful for those people who wanted to pursue their research and teaching or in higher education in the area of biotechnology. I just share my screen. Madam Devika, madam, you need to tell me whether my slides are visible. Hope my slides are visible to one and all.
Yes, sir.
Yes, sir. Yes, sir.
It is visible, sir.
Okay. Thank you. Thank you. So, in another 1 hour or so, I wish to tell you what are the career options in the area of biotechnology. Although the area of biotechnology is the new and emerging trend in the basic sciences. So, dear students, basic sciences play a pivotal role in the development of country's economy. If any country wanted to progress, or the progress of the nation is directly proportional to progress in the basic sciences. As long as we are interested in basic sciences, then only we can think of progress in the nation or progress of the country. One of the recent field in biotechnology, if you want to know the career options in biotechnology, you need to know what are the lengths and breadths of the subject. If you know the ins and outs of biotechnology, then only you can decide whether you are willing to pursue biotechnology as your career option or not. Mainly to know the importance of biotechnology, because without knowing the importance of biotechnology, you may not enjoy or you may not feel like taking biotechnology as your career. So if you want to ask biotechnology as your career, it is utmost important to know the importance of biotechnology. Or in other words, if you know the importance of biotechnology, you can decide very well whether you opt biotechnology as your career or not. So I will be dealing with what is the length and the breadth of biotechnology, and at the end of my slide, I will be giving you what are the agencies, either from the government or from the private sector, which includes the research in biotechnology. What are the government agencies which support the research in biotechnology? What are the private agencies which support the research in biotechnology? Although Government of India has various schemes to support the basic sciences and also to support the biotechnology. My dear young friends, please listen to me very carefully. Biotechnology is the only subject which has got a branch in the Ministry of Government of India. Under Ministry of Science and Technology, we have a separate discipline, division called Department of Biotechnology. No other subjects are having such a wonderful caliber. Either other science subjects or any other arts and humanities will not be having any in separate discipline under the Ministry of Science and Technology. This is the only subject which is finding its place under the Ministry of Science and Technology, Government of India. This indicates that what is the importance of this biotechnology. How you need to think of taking ahead of the subject of biotechnology. So, under Ministry of Science and Technology, we have a separate wing by name Department of Biotechnology. Their intention is to only support the programs related to biotechnology so that the research and development in the area of biotechnology will be continued, will be progress. As I said earlier, if you want to know the progress in the country, it is at most important to know the basic science research progress. So biotechnology has got such a wonderful offshoot of science and technology. So science and technology have been and would continue to be more vigorously and philosophically in the driving seat for rapid, sustainable socio-economic development in the world. So entire world, if you want to see the progress, the science and technology has the driving seat. Science and technology have been and would continue, not only in the past, but also in the present, but in future also, science and technology have been and would continue to be more vigorously and philosophically in the driving seat for what part? For rapid, sustainable, and socio-economic development in the world. That means if you want to see any rapid, sustainable socio-economic development, science and technology should be in the driving seat. Only the driving seat of science and technology will carry or will pull or push the country's progress or the progress in the world. Under this basic science and technology program, a new frontier of science, that is modern biology or biotechnology, the greatest intellectual enterprise of humankind. So I told you, under Government of India scheme, under Ministry of Science and Technology, we have a separate wing called Department of Biotechnology. Their intention is to only support the programs in the area of biotechnology. That is how the importance of this biotechnology is there. And biotechnology, as we all of us already know that, is a discipline which enables its exponents to convert raw materials to the final product, which either the raw materials and or a stage in the production process involve biological entities. So biological entities are playing a key role. Biological entities or microorganisms or plants or animals are playing a key role in the progress of new discipline of science and technology, that is biotechnology. And the business world defines this subject biotechnology as making money with biology. Look at the speaker of business world. How exactly those people who are involved in the business are calling this as making money with biology or making money out of biology. So biotechnology, bios means life, technology is soul, logos means study. Literally, it's the study of tools from living organisms. Biology, it's all about life. So life, so living things, plants, fungi, bacteria, viruses, animals, protists, all these things are included in this particular field by using those naturally available materials, whether you can think of improving the standard of life, the standard of life of human beings. Biotechnology combines the disciplines like all these things. So you have engineering in this biotechnology, you have computer science, you have cell and molecular biology, you have microbiology, you have genetics, physiology, biochemistry, immunology, virology. embryology, or DNA technology, and all these things, genetic manipulation of plants, genetic manipulation of bacteria, genetic manipulation of viruses, fungi, exclusively for the development of specific products which are beneficial for human beings. So what is biotechnology? Biotechnology is nothing but a collection of techniques starting from the scientific knowledge of cell biology, chemical engineering, biochemistry, biochemical engineering, microbiology, molecular biology, physiology, immunology, genetics, material science, computer science. All the scientific knowledge will be made utilized through biotechnological tools. Start from bioprocess technology, biosensor technology, tissue engineering, cell and tissue culture technology, genetic engineering, DNA chip technology, monoclonal antibodies, immunology, protein engineering. So what are the useful applications? There are three important fields of useful applications. That means all the useful applications under biotechnology can be studied under three important groups, whether it is green biotechnology, agricultural biotechnology, or environmental biotechnology, or red biotechnology, or medical biotechnology. Start from diagnostics, therapeutics, vaccines, under red biotechnology, environmental biotechnology, and agriculture biotechnology to improve the crop yield, food quality, and animal health. And dear friends, what is the role of biotechnology on this world is the basic information for that is according to United Nations Organization FAO, Food and Agriculture Organization, only 2 out of every 5 people are having the balanced 2 meals per day. And Malthusian theory you might have heard, global population is exploding in the geometric progression, whereas food production is increasing in the arithmetic progression. So what is geometric progression and what is arithmetic progression in the sense? Geometric progression will be encroaching the forest. That means in all these human activities, arable land is sinking day by day, whereas population is exploding. That means within the limited amount of reduced amount of land, you need to grow more because you have got a greater responsibility to feed millions and millions of hungry mouths. Agriculture land is decreasing, agriculture land is sinking on one hand, whereas population is increasing in the other hand. So whatever reduced agriculture land is there, within that reduced agriculture land, you have a greater responsibility to produce more because population is exploding. You have a greater responsibility to feed billions and billions of hungry mouths. So biotechnology for the millennium, these are all the different disciplines of biotechnology. Although all these disciplines can be studied in the four important categories: green biotechnology, red biotechnology, blue biotechnology, and white biotechnology. So glimpse of new millennium, the biotechnology for the new millennium, a few glimpses. It includes transgenome research, crop biotechnology and transgenic, brain research center, immunology, cell science, special societal programs from biotechnology, human resource development, you have aquaculture, you have environmental pollution abatement, that means how to control the environmental problems. All these, for all these problems, you have a solution from the field of biotechnology. You take any problems which are facing by humans, including nature, or food, or medicine, all these requirements of human beings, you have an answer in the field of biotechnology. So how? Let us see how. So I told you different types of biotechnology. Green biotechnology, which is agriculture biotechnology. Red biotechnology, which is medical biotechnology. Blue biotechnology, which is aquatic biotechnology. How to improve aquatic organisms, whether we can use them as consumption purpose or how to exploit the aquatic sources for the benefit of mankind. You have major industries of other, which is sourced from marine organisms, red algae, seaweed. So you need to collect them, process them, dry them and process them, then produce others. Then white biotechnology, that is industrial biotechnology. So there are four important types of biotechnology. So if you know at least few examples from each one of these fields, you will really enjoy biotechnology. Then you will make up your mind to opt biotechnology as your career program. Okay, at the end I tell you how to join this biotechnological course. But before that, you need to know the importance of different fields of biotechnology. If you know the importance of different fields of biotechnology, then only you will enjoy or you will address whether you will decide whether you will opt for biotechnology or not. So what is green biotechnology? Green biotechnology is nothing but plant biotechnology, which is commonly known as plant biotechnology, which apply to agricultural process to produce more environmentally friendly solutions, alternative to traditional industrial agriculture. So what are the drawbacks of uh, industrial agriculture? You put more chemical fertilizers or pesticides so that those chemicals will be there in the food chain. The residual effects of those chemicals will join in the food chain and somehow they reach the human body and responsible to cause hitherto not reported diseases. That is how our green biotechnology is coming into rescue of these problems. You might have read or you might have seen in the TV channel, there are areas of Kerala, Kasaragod, where the chemical pesticide has created almost all devastating problems to humankind. And to overcome those things, to overcome, to avoid the use of pesticides, insecticides, or fungicides, you can think of alternative from the field of green biotechnology. So what is the history of biotechnology? History of green biotechnology. Plant biotechnology is founded from the cell theory of Sleden and Strand. Students might have read the cell theory proposed by Sleden and Strand to biotech crops and the principles of cellular pluripotency and the genetic information in the bacteria by Frederick Jeffrey. So pluripotency is the inherent capacity of the cell to give rise to an individual part or individual entire organism when it is being provided to the suitable atmospheric conditions. If the cells are pluripotent, then only it will give us the complete plant. Of course, you need to provide the suitable environmental conditions. So what are the aims of green biotechnology? To tackle the food security issues or how to improve the growth of the plants, how to improve the fuel and reduce the environmental footprint of the industry, carbon footprint, the environmental problems. You might have read or you might have seen in the television few years back or last year, the schools in Delhi were set for almost 1 week or 10 days or 15 days because of the greater environmental pollution problems. Because we have been utilizing these fossil fuels, we have been utilizing the chemical pesticides, all these things create a problem in the environment so that you cannot see the concentration of particulate matter in the air. So agricultural biotechnology or the green biotechnology. So what is the history of crop improvement? By trial and error for almost 9,900 years, almost 10,000 years, people have tried based on trial and error to improve the varieties of the plants for the consumption of food by scientific principles of breeding for last 100 years. So biotechnology or agriculture biotechnology by using scientific principles of plant breeding since the last 100 years or so. By chemical induced mutation for last 75 years and by recombinant DNA technology for last 25 years. That means how exactly the biotechnological tools coming into help rescue of the farmers or to address many of the environmental problems. So what are the old and new approaches of plant improvement? Traditionally, old approaches you need to keep on breeding using crossing with different plants, different species of plants or different varieties of the plants to get new variety with the desired size or desired characteristic features. But the biotechnological tool helps us to identify a desired gene and try to transfer the desired gene into a commercial variety so that new variety you will be developing along with the desired gene. So the traditional plant breeding will take almost 10 years to come to arrive at one particular variety. Whereas within 1 year or so, you can reach based on the biotechnological tools and applications. So what is coming that will have benefits and loss also, both for food security and health. We have the examples of drought tolerant maize. We have the examples of crops suitable for industrial applications like biofuels or the plastics, modified cassava and the cowpea, health benefits in the oil crops, and allergen-free peanuts. So all these things will be possible only when you use the tools and techniques of biotechnology. So based on the available tools and techniques, you can improve the crop. Crop improvement can be done. You can improve the crop based on your required traits or genes. So tailor-made crops or pharmaceuticals or custom designed crops can be you can think of using plant biotechnological applications. So biotechnology of plants and specific crops including genetically engineered organisms for yield and quality traits or their metabolite production, which are rapid clonal propagation and germplasm conservation. You have many problems. Every now and then some plant species will be disappearing, endangered. So using the cell and tissue culture of biotechnology, you can think of maintaining or conservation of that particular endangered plant species. So gene transfer to enhance the agricultural productivity. So you need to identify the gene which is beneficial or which is the character which human wanted or beneficial to humankind. So what are the new approaches? Agriculture biotechnology, the agriculture production has more than doubled while the amount of land under cultivation has actually declined. So as I said earlier, within the available limited land resources, you can think of growing more, producing more, because you have a great responsibility to feed millions and millions of hungry mouths. Most direct way to use biotechnology to improve the crop agriculture is the genetically engineered plants. That is, one can alter the base of the genetic structure, whether it is in terms of high yielding, whether it is in terms of disease resistance, whether it is in terms of implementing the medical property or medicinal property of the particular plant. All these things are possible only by using cell and tissue culture technology. So recombinant DNA technology, application of molecular markers, tissue culture technique, and recently, of late, one more technique has been introduced or included into the list, that is RNAi technology, RNA interference technique. So what are the different mechanisms of including all these things? Recombinant DNA technology, if you take, plants have been rendered resistant or in terms of high yielding to the specific pathogens by genetically engineered, transforming them with isolated specific genes that provide resistance against these pathogens. Those plants are called as transgenic plants. And of course, you have drawbacks of this also. Altering the genes in plants to fight pests and improve the nutrition. You can think of whatever desired traits you wanted to have it in tomato plant. You wanted to have in banana fruit. That you can do by using plant biotechnological techniques and tools. So what are the basic steps? Isolate the gene of interest, vector is chosen, and all these technical details students you can study when you are pursuing your education in the higher classes. So this is how the gene transfer in plants will be taken place. And you need to identify the first what gene you want to transfer. When plasmid is removed from bacterium and the tDNA is cut by a restriction enzyme. So these are all the important steps. Foreign DNA is cut by the same enzyme. The foreign DNA is inserted into the tDNA of the plasmid so that you will get recombinant plasmid, and from that recombinant plasmid is reinserted into a bacterium, and those bacterium is used to insert tDNA carrying the foreign gene into the chromosome of a plant. Plant cell, subsequently, if you can culture those plants, it will have the genes which you have transferred or gene of interest. So whatever gene of interest, either in terms of disease resistant or high yielding, all those characteristic features are there in this transgenic plant. So the best example of this is the biotechnology product where FAO is recommended to all underdeveloped nations. The golden rice is the best example where FAO is recommended to all underdeveloped nations to take care of vitamin A deficiency disease or vitamin A deficiency syndrome, night blindness. So those people who are suffering from night blindness, if they can have the golden rice, that night blindness to some extent it will be eliminated. But the mothers, the pregnant women, if they have the golden rice, the kids born from that particular woman will be free from vitamin A deficiency. So what they have done here is that they have isolated the genes from two important sources. The first one is the flowering plant, that is daffodil, and the second one is the bacteria, that is Irvinia. So genes from the daffodils and the genes from the Irvinia have been isolated and transferred through agrobacterium mediated gene transfer. Then they have been inserted into rice plant, and this transgenic rice plant showing provitamin A rich. So if you consume that golden rice, this particular provitamin A rich variety, that will take care of your vitamin A deficiency. And remember, this golden rice is the product which is recommended from FAO to the underdeveloped nations or developing nations to get rid of, to take care of, to alleviate night blindness disease, that is vitamin A deficiency syndrome. So what are the different biotech crops? Enhanced protein and essential nutrients to prevent disease, vitamin A to prevent childhood blindness, increased calories and nutrients to prevent malnutrition, increasing food availability by reducing the spoilage. You can reduce the spoilage because these are the experiments in some vegetable plants like tomato, where you can enhance the keeping quality or the shelf life of that particular vegetable. So nutritional quality, if you take golden rice is the best example, expression of enzymes of beta carotene, phosphate in rice and endosperm. That is mainly to ameliorate vitamin A deficiency. Then the other aspect is nutraceuticals. I was telling you about tailor-made food, custom designed food. Whatever nutrition you want to have, say for example, whatever vaccine you want to have in terms of banana, or whatever action you want to have in terms of any other fruit, apple, or rice, or wheat. So added nutrients, either in terms of pharmaceuticals or in terms of vaccines, any of these things you can think of adding to whatever crops, whatever foods we eat. So the other thing is fighting hunger, improving the yields of food staples, controlling the insects, controlling their diseases. The best proven examples are banana, cassava, and sweet potato in case of virus, and greater salt tolerance. So you can think of growing the plants with high quality, growing the plants use of less pesticides, less insecticides, and less fungicides. So you can also achieve food security, increasing the food productivity or crop productivity to meet growing global food needs. I told you, global population is exploding in the geometric progression, whereas food production is increasing in the arithmetic progression. You need to have a great responsibility to produce more in the less amount of arable land. And we are losing arable land day by day by various anthropogenic activities, by industrialization, by urbanization, and for construction of roads. For all these things, we are losing day by day the arable land. Generating high yielding varieties. These are some of the examples of host plants. Several parameters such as plant height, tiller number, the photoperiod, insensitivity, like rice, arabidopsis, barley, all these plants have been already the successful story. Then what is the importance of biotechnology in the environmental field? Environmental biotechnology. Apart from pest resistance, I told you, if you take Government of India statistics, 60% of the produced pesticide is taking only to control one particular disease in one particular crop, and that crop is being grown only in the 8% of the land. This is the statistic. For example, cotton crop. Cotton crop is being grown only in the 8% of the total agriculture land in India, but 60% of the total pesticide produced from Government of India, you need to use only to the cotton crop. You can just imagine the amount of pesticides which is being used for the cultivation of the crop species. Biotechnology uses an innovative and eco-friendly approach to improve pest control management. Although there are various pros and cons, I'm not going to debate whether this is good or this is bad or this is not good, something like that. I'm telling you the basic information for the purpose of academic purpose to the young minds of BSc students. About 40 genes obtained from microorganisms of higher plants and animals used to provide insect resistance in crop plants. Certain genes from higher plants synthesize products possessing insecticidal activity. This is the basic point which you need to understand. The basic point is certain genes from higher plants synthesize the products possessing insecticidal activity. The copy trypsin inhibitor gene is the best example. It can be think of transferring to tobacco, potato, and oilseeds insecticidal protein. A trypsin inhibitor that was capable of destroying insects belonging to the order Lepidoptera, Coleoptera, Orthoptera, etc. The first gene available for genetic engineering of crop plants for particularly pest resistance were cry proteins from Bacillus thuringiensis, a soil-borne bacterium. The most notable example is BT cotton, where the crop is the success story contains a Tri1AC gene that is resistant to a notorious insect problem that is Helicoverpa ornithira, Helicoverpa species. Transgenic crops, that is bulbworm, transgenic crops with BT genes, for example, cotton, rice, maize, potato, tomato, brinjal, cauliflower, cabbage, etc. have been developed already and being used in commercial market. So basic crops protect plants against the specific insect pest. This is how the insect pest is attacking to the cotton crop. The unique feature of this insect disease causing organism, Bacillus thuringiensis, is the production of crystal-like proteins that selectively kills only specific groups of insects. It is not killing uniformly across the species. So when the insect eats these proteins, its own digestive enzymes activate the toxin form of the protein. And proteins bind to specific receptors on the intestinal walls and rupture the midgut cells and thereby killing the insect. This is how the mechanism of BT cotton working. So BT cotton is the first genetically modified crop in the year 2002, second in the global cotton production, and the other things. So these are the list of proteins, crystal protein of Bacillus thuringiensis, and what is their specificity. That means to what groups of organisms they are active against. Either they are active against Lepidoptera or Coleoptera or Diptera, etc. Transgenic plants expressing the foreign gene for insect resistance, whether cotton or brinjal or maize or rice. What is the foreign gene which is being used in the cotton crop? What is the foreign gene which is being used in the brinjal? All these things are given here. Whether the origin of the gene, whether it is from Bacillus thuringiensis or corn or potato, and targeting insect pest is also being given. So against this particular insect pest, this tri-gene is working video cover for J or other insect organisms. So these are the crops again, the list of foreign gene or the origin of the genes and the target insect pest. BT crops protect plants against specific insect crops, even in BT maize. BT maize also will be it has released and it will be taken care of most of the insect and the fungal disease problems. And this is how the people are spraying insecticides by creating a lot of pollution to the environment. By spraying this insecticide to cause lot of pollution. Sometimes if you keep on spraying, the target organism is also developing resistance. And the other problem is the residue which is there after reaching the target organism, if at all if it is there in the residue, by any chance if it reaches to the human being through food chain, it will cause hitherto not reported diseases. So brinjal is also there, and this is how putting the science into agriculture, different shapes, different colors, different sizes of brinjal. So what is the result of biotechnology? An organism showing a novel type not normally found in the species. For example, a flavor sour tomato mainly to increase the keeping quality of the tomato. Extended shelf life. We have seen most of the farmers in the area of polar and other regions of Karnataka state, they dump the tomatoes on the road because of reduced price. So once if you grow, within a week's time or within 10 days time, it will be reached to the end user. If it is not reaching, then the entire crop is at loss. Entire crop is at loss because reduced shelf life of those plants are there. By any chance, by using this so-called techniques in biotechnology, if we can extend the shelf life, if you can increase the keeping quality, you can reach the farmers or you can reach the consumers even after 1 month of time. So herbicide tolerance is another field. Weeds are unwanted plants which decrease the crop yield by competing with the crop plants for light, water, and the nutrients. Several biotechnological strategies for weed control are being used. Overexpression of the target protein to permit normal metabolism. And the best example is Roundup Ready. It is a pesticide or the chemical which you can spray crop plants tolerant to this particular pesticide because it will kill only the weeds. Herbicide is weedicide. Herbs are the small organisms and those Roundup Ready, if you spray the farmer spray, it will kill only the weeds and it will not do any harm to the crop plant. So herbicide resistant plant is there. Glyphosate Roundup Ready is environmentally safe herbicide. Indefinitely kills the crops as well as the weeds. Otherwise, scientists have isolated the glyphosate resistant gene and successfully transferred it into cotton, soybean, tobacco, and tomato. So that if you use Roundup Ready, it will not kill the crop plants, it will kill only the herbs or the weeds. Gene was derived from this particular bacterium, Salmonella typhimurium. Genetic engineering for crop protection, because if you can reduce the use of chemical fungicides, you can do these biotech related crops, the resultant products of the crop. Production of crops from pests, insects, viruses, bacteria, nematodes, fungi, and weeds. Chemical pesticides, herbicides are not selective enough to affect only harmful organisms. They affect even humankind. New approaches to animal agriculture or animal breeding, fish farming, all these things are the resultant products of so-called biotechnology. Genetic engineering for transgenic crops overcomes the limitations of traditional breeding, allows scientists to use new traits from many kinds of plants and other living organisms. So generating high-yielding varieties by genetic manipulation of plant architecture. The father of green revolution, Professor M.S. Swaminathan, is World Food Prize winner who is responsible for increase the productivity of crop plants. Of course, he has not used any genetically modified organisms. He has used semi-dwarf wheat varieties to produce more. Green Revolution, Norman Bobag and Professor M.S. Swaminathan with irrigation facilities, improved hybrid seeds, chemical fertilizers, pest management, all these things result into the self-sufficiency of Indian crop productivity. So application of GM technology. So yield, nutritional improvement, all these things are the applications of genetically modified organisms technology. You can think of producing edible vaccines also, herbicide tolerance, and vegetables. These are the examples of the transgenic crops of food, fruits, edible oils, cereals, and legumes. All these things you can uh, take the information. And biofarming. Biofarming is growing transgenic crops that express pharmaceutical products. For example, drugs, antibodies, or the proteins. By any chance, if you consume one banana along with this pharmaceutical product, you need not have to take any other vaccine or drug for a particular disease. Like that, you can think of producing tailor-made products, tailor-made plants, tailor-made custom-designed crop species. Edible vaccines, a biofarming gene, biotech plant serving human health needs. A pattern protein gene is enclosed and gene is inserted into the DNA of the plant, including potato, banana, tomato. Humans eat that particular plant so that the body produces antibodies against that particular pattern protein. Humans are immunized or vaccinated. The examples which are already available in the market are hepatitis B, diarrhea, measles, etc. So future health-related biotech products. You can think of developing vaccines for herpes, hepatitis, AIDS, and malaria, including this growth this year also. So agencies of public sector promoting the agricultural biotechnology. I told you, Ministry of Science and Technology has got a separate wing for Department of Biotechnology, and which will support all the programs of biotech related research, either in the science stream course, regular course, degree course, or master degree course, or the research in the field of biotechnology. And Department of Scientific and Industrial Research, that is Council of Scientific and CSIR, and Ministry of Agriculture, all these are government agencies which support the biotech program. And there are various private organizations which are supporting the transgenic crops. International Center for Genetic Engineering and Biotechnology, public sectors, all these are public sectors again. These are the private sector, the private companies. And target crops are transgenic research in India. Cereals, if you take rice, wheat, maize are there. Vegetables, you can think of brinjal, tomato, etc. And target rice, disease resistance or improving quality of the protein, increasing vitamin content, stress tolerance, herbicide resistance, etc. And what are the environmental impacts and social responses to genetically engineered crops? So potential benefits of transgenic organisms which are environmentally safe, healthy in terms of social acceptance. But only thing is you need to educate the people to use this is safe and you can use so that you can reduce so much of amount of pesticide, insecticide, fungicide, etc. So genetic engineering provides a greater range of possibilities for transgenic transferring desired traits into the organism. The potentially biological novelty, those are novel organisms. A greater diversity of the organisms may be modified, and the quality and the quantity of traits are limited by the identification of useful genes which are not constrained by existing variation among interbreeding relatives. Are they good or bad to the environment? Both opinions are there. Some people say it is good and some people say it is not good. According to the plant medicine industry, they are good. But according to environmental activist group, they say it is not good. But whatever all said and done, whether it is good and bad, biotech industry is picking up and they are ready to accept whatever the product which is being used commercially. Scope of genetically modified crops. These are disease resistance, herbicide, all these things. So what are the genetic modified organisms which are already there in the commercial? Of course, these are all not in India. These are only in USA, UK, and the countries like that. Biotech benefits and risks, both benefits and risks are also there. And the expected biotech impact areas and the side. These are, if you say basic science, biotech application in the field of health, in the field of food and feed, in the field of environment, in the field of industrial, and in the field of primary productivity. I told you there are the branches of biotechnology like red biotechnology, green biotechnology, white biotechnology, and blue biotechnology also. For a clean environment, you need to have the biotechnology with the power of microorganisms. The superbug is the best example. Anand Chakravarti's work, best example. It will take care of your most of the oil spills, environmental cleaning. Biotechnology helps to eliminate pollutants, removes waste from the environment. And the next is red biotechnology. Red is medical. This term is used in medical process. It involves the manipulation of microorganisms to make products such as antibiotics. So microorganisms are omnipresent, they present everywhere. Once if you inhale, you will inhale millions and millions of microorganisms. But once if you exhale, same amount of microbes are coming out from your body. Our GI tract harbors almost 600,000 different types of bacteria. Our GI tract, gastrointestinal tract, harbors 600,000 different types of microorganisms. So it possesses all the acidic pH, alkaline pH, neutral pH. So based on the pH, the population of organisms will be discriminated. In acidic pH, you have microorganisms. In the neutral pH, you have microorganisms. In the alkaline pH, you have microorganisms. So red biotechnology includes production of medicines and pharmaceutical products for treating or diagnosing diseases or disorders, designing of organisms to manufacture antibiotics and vaccines, engineering of genetic defects through genomics of manipulation, use in the forensic through DNA profiling. Because although we are all human beings, but our individual fingerprints are not exactly similar. That is the sequence of DNA which is present in each and every cell of an organism, not only in human beings, but also in the plant. Examples: production of human insulin from non-human sources, production of hormones like interferons, cytokinins, steroids, human growth hormones. So gene therapy for prevention and control of diseases like hemophilia, cystic fibrosis, development of vaccines and antibodies for rabies, HIV. All these things are there, my dear friends. Whatever interest you have got, either you have got interest in red biotechnology, you can pursue your career in the red biotechnology. If you are interested in green biotechnology, you can pursue your career in the green biotechnology. Similarly, blue biotechnology and white biotechnology also. Development of medicine with the help of biotechnology. Microorganisms can be used to make biological substances for humans which are equally effective as natural substances. So treatment of diseases like hepatitis and arthritis are made possible with the help of the red biotechnology. Insulin is the best example. Human insulin is being produced using genetically engineered organisms such as humulin. The name of the product is humulin and is used for the treatment of diabetes, that is low sugar level in the blood. Interferon is the other example. Interferons interfere in transmission of viral genome from one cell to another, and it also inhibits the cell division of the abnormal cell. Interferons produced using recombinant DNA technology is used to treat the cancer patient. And the next is blue biotechnology. Blue biotechnology is the application of molecular biological methods to the marine and freshwater organisms. Oceanographic Institute is there in Goa, National Oceanographic Institute, National Institute of Oceanography, NIO. There they are doing using biotechnology for the improvement of marine and freshwater organisms. So important marine forces and research areas in the field of food, in the field of energy, in the field of health, in the field of environment from the marine sources. Algae are invertebrate fishes, and the aim is to develop innovative methods to increase aquaculture production, zero waste recirculation systems. So all these things, the entire table shows the important marine forces and research areas, those who are interested in blue biotechnology. Algae is the best example. You can think of improving the algae for the consumption. There are many products which are already available in the market where people are consuming, like single cell protein, SCP, marine biotechnology securing alternative source of renewable energy, production of biofuel from the microalgae, production of oil and fat from microalgae. Research priorities to improve microbial enhanced oil recovery from the marine organisms. Marine best example is marine biotechnology securing human health. Best examples are marine derived anti-cancer drugs, marine derived painkillers, marine derived antibiotics, and marine derived cosmetics. In all these fields, blue biotechnology is important. Application of blue biotechnology, these are various fields you can think of to improve aquaculture, transgenic of marine organisms, disease resistance, conservation, seaweeds and their products. You can improve the pharmaceuticals, you can improve the production of enzymes, you can improve the biomolecules, or bioremediation is also the use of microorganisms to alleviate the problems of pollution. Seaweeds and their products. I told you nutraceuticals, SCP, single cell proteins, chlorella, biodiesel, and bioremediation, pollution indicator, all these things you can think of from the blue biotechnology. I am just giving you just a drop in the ocean, tip of the eye, and there are ocean amount of information, large amount of information in all the fields, all the four fields of biotechnology. You can think of pursuing your career either in the green biotechnology or blue biotechnology or red biotechnology or white biotechnology or whatever. So industrial biotechnology, it is often referred to as white biotechnology, is a collection of scientific techniques and technologies used to improve both the efficiency and environmental footprint of modern industrial production. You can think of producing any of the organisms or any of the organism's products for the benefit of mankind. Agar is one such example, obtained from jittery plants, Viridium gigatina gracilaria. And you can name many different organisms sources for consumption of human beings. Microbial technology constitutes the core of industrial biotechnology. The production of alcohol from the event manager. Event manager is one particular fungal microorganism, one particular fungus which is managing the entire flow. Microbial technology refers to the use of microbes to obtain the product of service of economic value. It is also called as the process of fermentation. Activities of industrial biotechnology. So what are the activities which are present in the industrial biotechnology? Isolation of microorganisms from nature, their screening for the product formation, improvement of the product yield, maintenance of the culture, and mass culturing using bioreactors or fermenters. And you can recover them from those fermenters. Application is metabolite production for the benefit of mankind, either in terms of medicine or in terms of food or whatever. The anaerobic digestion for methane production, waste treatment both for organic and industrial waste, production of biocontrol agents. This is one important product in the area of agriculture biotechnology. Fermentation of food products. You can think of producing the fermented food products from the otherwise unaccepted plants and the other food items. Bio-based fuel and the energy. Metabolite production like acetone, butanol, alcohol, antibiotics, enzymes, etc. All these things you can think of improving with the help of biotechnology. Biomass production, microorganisms employed to enhance the availability of nutrients, nitrogen by fixing atmospheric nitrogen, and phosphorus by solubilizing soil phosphorus to the crop. Those are called as biofertilizers. Example, azolla, azospirillum, rhizobium, etc. Recovery of the metals, bioleaching is another field. You can think of using microorganisms to the process of recovery of metals. Microbes can be employed to recover valuable metals from the low-grade ores and also from the dilute solutions for which the conventional metallurgical processes are uneconomical or rather generally rather polluting. Leaching of copper as copper sulfate from the ore, the best example. Production of biocontrol agents, trichoderma or bioinsecticides or bioherbicides, all these things. Then fermentation of food products. You can think of the fermentation of the cabbage, fermentation of tomato, fermentation of chili. Otherwise, you can the shelf life of those things are very low. If you subject them for the process of fermentation, you can increase the shelf life or the keeping qualities of those food products. In some cases, it is improving the nutritional quality. In some cases, it is not improving the nutritional quality, but it will improve the keeping quality or the shelf life of those products. Bio-based fuel of the energy. Ethanol is currently produced by fermentation of grain. That is old technology. You can think of using newer technology from the biotechnology to produce bio-based fuel and the energy. Benefits, these are all benefits. Low input of substrate, high rate of output, friendly to the environment, renewable, increased efficiency, all these things. Then so this is in brief. I told you this is just a drop of ocean, the importance of biotechnology and all the fields of biotechnology. There are four important fields and all the fields have just made an attempt to touch, not gone into detail. Now comes exact picture, that is career options with an Indian context. Normally we take 10 plus 2, pre-university course, pre-university course as starting point. Actually, from that point, if you take whatever 10 plus 2 you score from the basic sciences, if you score high amount, say for example 90% and above into pre-university, and not gone to either medicine or engineering, but if you are pursuing your education in the basic sciences, you will have the scholarship from DSC under the KVCY program, Kishore Vijnana Puraskara Yojana. Mysore Vaidyanik Puraskar Yojana from DSP operating from Indian Institute of Science, Bangalore. They will conduct a test, you need to qualify in the test. So that if you score more in pre-university And if you are not joining to either medicine or engineering, and if you are pursuing your education in basic sciences, Government of India will support by giving 2,000 rupees every month fellowship. So, for example, you have scored more than 90% in the pre-university UC course, and you did not opt for either engineering or medicine, but you have opted for basic sciences. Government of India will fund for the entire education, and this is the entire picture. So, if you complete 10 plus 2 pre-university, these are all the courses like science, arts, commerce, diploma, or any other training courses. But if you choose science as your career, you need to appear for these examinations. Either KVPY, Kishore Vaidmanika Praskara Yojana, JEE, NTA, NDA, IAC is also conducting the entrance test. You can, if you clear this IAC entrance test and KVPY, if you pursue your studies in the basic sciences, Government of India will give you fellowship. And of course, these are all other engineering, medicine, agriculture, horticulture, all these things possible. Then if you complete graduation by qualifying KVPY, you will get a fellowship from all the 3 years BSc. And during the BSc course, again, if you can appear for entrance examination, like GAT-B examination, Graduate Aptitude Test for Biotechnology. If you want to pursue your education in biotechnology, again for your MSc course, Government of India will give you the money. The other way, even if you pursue your agriculture, horticulture, or fisheries, but if you want to pursue your education in biotechnology, then you have to appear for GAT-B examination. Then Government of India will give you the money, the fellowship. So those students, if you are interested to pursue your MSc in biotechnology, please appear for this examination of GAT-B, Graduate Aptitude Test for Biotechnology. And in the month of July, that is in the next month, Government of India will make advertisement. So please keep a watch on the internet of DBT Government of India website. They will call for those GAT-B examination, those who are interested in pursuit of MSc Biotechnology, you will get the fellowship of 5,000 rupees every month for MSc Biotechnology program. And even in Mysore, if you study in this department, you will be given 5,000 rupees fellowship every month for 2 years, 24 months. Either you colleges may run or may not run, or online classes or offline classes or whatever, you will get fellowship. But you need to clear this GAT-B examination. And of course, IAC is also conducting online examination or conducting the entrance test for integrated PhD program. Both MSc and PhD you can do by clearing that IAC examination. And similarly, you have many other examinations just like any other degree, like all UPSC, Union Public Service Commission, UPSC examination, BSRB, RRB, Railway Recruitment Board, Staff Selection Commission, all these things you can appear. Even if you are a life sciences graduate, you can appear all these examinations. But if you want to pursue your education, if you want to pursue your master's in MSc Biotechnology, you please appear for this examination of GAT-B, Graduate Aptitude Test for Biotechnology, which Government of India is conducting. So, Government of India, Regional Center for Biotechnology, Faridabad is responsible for conducting the examination, and it is just online examination. Last year I told I have gone to some other colleges and requested them to appear, and some people have qualified and they have joined this department. I am very happy to share this information. They are getting the 5,000 rupees scholarship every month. Since I am the coordinator for that MSc Biotechnology program, I request all of you to appear for this GAT-B examination to pursue your studies of MSc Biotechnology. And once if you are doing MSc Biotechnology, you are eligible to appear for all these examinations. UGC CSIR, University Grants Commission, Council of Scientific and Industrial Research, Junior Research Fellow, UGC CSIR JRF. DBT is also conducting a separate test as DBT JRF. This is exclusively for MSc Biotechnology students. This is for all life sciences students. MSc Biochemistry, MSc Botany, MSc Zoology, MSc Agriculture, MSc Microbiology, MSc Biotechnology, Biochemistry, all these things can appear. But for this DBT JRF, only MSc Biotechnology students are eligible to appear. Then ICMR JRF, ICAR JRF, you can do all these things if you want to pursue your research or PhD in India. And of course, if you want to pursue a PhD abroad, graduate recording examination, test test to English as foreign language, that is GRE TOEFL examination. And if you complete your PhD, sometimes you can think of going for abroad as postdoctoral fellowship, postdoctoral fellow, PDF. And if you do not want to go to abroad, you have 40,000 first grade colleges in India, 40,000 first grade colleges which are offering MSc Biotechnology, BSc Biotechnology course, where you can think of becoming assistant professor. And 875 state universities and some 70 central universities, almost 1000 central, 900 central universities. And you have number of research institutes where you can submit your resume for the position, which I'll be showing you in the next slide. So I once again request all BSc graduates who are interested in MSc Biotechnology, please look for this notification of GATB, Graduate Aptitude Test for Biotechnology. If you qualify this test, you are entitled to receive 5,000 rupees fellowship every month for 24 months. And this department is also one of the recognized center to offer this MSc Biotechnology course. It is DBT recognized. About 15 candidates you can take the admission from GATB qualified, and of course we have university entrance test also to join for MSc Biotechnology. Then what are the different research institutes along with your universities? I told you about 40,000 first year colleges in India, and about 900 universities are there, along with number of research institutes including Mysore. This slide shows you some of the research institutes where you can think of joining for either for your PhD or as a scientist course. If you take from this corner ICGEB, these are all the institutes which are located in Delhi. ICGEB is International Center for Genetic Engineering and Biotechnology, New Delhi. Indian Agriculture Research Institute, New Delhi. National Bureau of Transgenic Resources, New Delhi. All India Institute of Medical Sciences, New Delhi. National Institute of Immunology, New Delhi. All these are research institutes which biotechnology MSc qualified candidates, either DBT entrance test or CSIR entrance test, are eligible to join for their PhD program. And in addition to that, some of the institutes like ICTB, International Center for Genetic Engineering and Biotechnology, NIN, and IASC, or National Institute of Nutrition, CCMB, CDFD, they conduct their own entrance test to take their PhD students. MSc Biotechnology completed students are eligible. Then comes Lucknow, NBRI, National Botanical Research Institute, Lucknow, Industrial Toxicology Research Center, Lucknow, Central Institute of Medicinal and Aromatic Plants, Central Institute of Medicinal and Aromatic Plants, Lucknow. These are all research institutes, CSIR labs located in Lucknow, where you can think of joining for your PhD and subsequently as a scientist course once if you complete your MSc Biotechnology and clear UGC, CSIR or DBT entrance test. Then BRC, Baba Atomic Research Center, Mumbai, NCL Pune, National Chemical Laboratories, Pune, NCCS Pune, National Center for Cell Sciences. These are also located research institutes located in Pune and BRC in Mumbai. Then these three things I need not have to emphasize: CSTRA, DFRL, CSR, and TI. These research centers are located in Mysore. These research institutes are located in Mysore, Central Food Technological Research Institute, Defense uh, Food Research Laboratory, CSR and TI, Central Agriculture Research and Training Institute. These three research institutes are located in Mysore. DKVK, Bangalore, Gandhi Krishi Vijnana Kendra, and Indian Institute of Science and some of IITs. Throughout India, IITs are located, but Indian Institute of Science is located in Bangalore. So GKVK is University of Agricultural Sciences, Gandhi Krishi Vijnana Kendra and IISc are located in Bangalore. IISR, Indian Institute of Spice Research, Kasargod. Then Central Leather Research Institute, Chennai. NIN, Hyderabad. NIN is National Institute of Nutrition, Hyderabad. CCMB, Hyderabad, Center for Cellular and Molecular Biology. Center for DNA Fingerprinting and Diagnostics. All these three institutes are located in Hyderabad. Even if you do your MSc in Biotechnology and clear UGC, CSIR and DBT, you can think of joining these research institutes. Then CPRI, Simla, Central Potato Research Institute. This is CRRI, Central Rice Research Institute, Cuttack. Central Rice Research Institute, Cuttack, Orissa. Then regional Rajiv Gandhi Center for Biotechnology, Faridabad, Delhi. Then INTEC, Institute of Microbial Technology, Chandigarh. Then CPRI, this is Kufri. Kufri means Simla, Central Potato Research Institute. These are only some of the very few research institutes, although there are thousands of research institutes funded from ICR, CSIR, ICMR, all these things, but I am just giving you whatever research institutes which our students have visited. I have taken my MSc students to these institutes for their educational trip, and only those institutes I have put, but because there are thousands of different research institutes where you can think of joining after qualifying UGC CSI after completion of MSc Biotechnology. Then some of the international research institutes, some of them are located in India, some of them are located in USA, some of them are located in Germany, Japan, all these things. For example, International Crops Research Institute for Semi-Arid Tropics. This is located in Hyderabad, Patancheru, Hyderabad. This is Mexico, Central Maize Research Institute. This is IRRI, International Rice Research Institute, Philippines, Manila. United States Department of Agriculture, Pasteur Institute. Then JSPS, Japanese Society for Promotion of Science. Then International Bureau of Transgenic Resources. These are some of the research institutes. Alexander Van Humboldt, DAAD Germany, DANIDA Denmark, DGSP Denmark, uh, Ford Foundation USA, Commonwealth Fellowship UK. All these are research institutes along with the research agencies, fellowship agencies where you can think of joining or pursuing your PhD either within India or abroad. This is, dear friends, this is in brief. I have given you only glimpses, only a few, very few points. If you want to know anything in detail, feel free to ask me. Either you can come and visit this department, or you can send me an email, or you can text me. Whatever ways and means, that should not be a barrier. Thank you. By this, I will stop at this stage. Thank you very much. If you have any questions or any clarifications, feel free to ask me.
Now the session is open for discussion. You may ask questions now. Please unmute yourself and ask. Is there anybody who wants to ask any question regarding this session?
But please remember, even if you do not ask any question, without forget to please appear for GAT-B examination. This is applicable to only final year BSc students. I request all the final year BSc students, please appear for GAT-B examination, which will DBT will notify in the next month, in the month of July.
Thank you, sir, for your wonderful talk. It was inspiration for the career in biotechnology.
Hello.
And now I call up.
Somebody has got some question, it seems.
Somebody is having a query, I think.
So thank you so much for sharing your profound knowledge on in biotechnology. And I request you to, can you please share the number of seats which is available in University of Mysore for the biotechnology students who are pursuing the BSc in different colleges in university, in the affiliated colleges in University of Mysore.
Okay. Thank you. Actually, I told you, those students who are studying in the final year BSc, please appear for GAT-B examination. There are 65 different universities which are offering MSc Biotechnology under DBT recognition. But each university will be having its own limited seat number. Only 15 seats we can accommodate. 15 seats of GAT-B exam qualified students. In addition to these 15, our University of Mysore quota is also there. Totally, we have 50 seats. Both Scheme A, Scheme B, there are various. In the university quota, you have different category-wise reservations, and Scheme A reservation, Scheme B, all these things are there. But those students who are joining under university entrance test will not be getting any of the scholarships. Thus, they need to pay the fees to the university. But if you are clearing, if you are eligible, if you qualify in the GATB examination, you need not have to pay anything. Even if you pay, that will be reimbursed from the DBT Government of India, and each university will be having their own limited seats. Some of the universities have taken only 10, we have taken 15. In addition to this 15 number, which is eligible for GATB qualified candidates, we have other 30 or 35, something like that. Totally it will come around 50, 51, something like that, because you have ICCR quota. ICCR means Indian Council for Cultural Relationship. Indian Council for Cultural Relationship. So some of the students who are from background countries like underdeveloped or developing countries, if they approach Government of India, they will allot simply whatever university that particular student is asking, like Sri Lanka, Bangladesh, Nepal, South African countries. These countries simply submit their application. For them, we cannot say seats are full. It is over and above supernumerary quota we call. But University of Mysore has got its own entrance, and based on the eligible marks, based on the merit, we allot the seats. Of course, you have many kinds of reservations. You have separate quota for NCC, separate quota for NSS, separate quota for sports and culture, separate quota for all these category of people. But only 15 seats for GATB exam qualified. Those candidates will be getting 5,000 rupees scholarship every month. I told you, even if you are presenting offline classes or online classes, totally you will get 24 months, 24 into 5,000 rupees every month.
Hello, good evening, sir.
Good evening.
Sir, what about students pursuing BTech in biotechnology, sir?
They
I mean,
are,
what kind of exams they can take?
they are also eligible for all these examinations. I have not emphasized that whatever bachelor's degree in biotechnology, either BE biotechnology or BTech biotechnology, they are also eligible to take these examinations. And remember, our MSc biotechnology is open to all science streams. You need not have to study only CPZ combination of BSc. You can study any combination in BSc. You can study dental, you can study engineering in biotechnology, BTech in biotechnology, medicine. All these students are eligible to apply for our course, provided they have to qualify entrance examination, and also they have to qualify GATB examination. I told you, if you qualify in the GATB examination, you are eligible to receive fellowship. But if you are not qualified in GATB, but you have to qualify in our own university entrance examination, for it you will not get any of the fellowship. Only GATB qualified candidates are eligible to get the fellowship.
Yeah, thank you, sir.
Tanushree, continue with the program.
Yes, ma'am. Now I call upon Tejaswini to propose the vote of thanks.
Good evening, everyone. It's my privilege that I have been given an opportunity to propose vote of thanks in today's session. First, I would like to thank our today's guest, Dr. Umesha, for his wonderful talk, which has given certain interesting facts and also helped us to know about our future career in the field of biotechnology. Once again, thank you, sir, for making some free time in your busy schedule and giving us this special talk. Next, I would like to thank our beloved principal, Dr. M. Devika, for encouraging and supporting us through this occasion and making this program a grand success. Once again, thank you, ma'am. Next, I would like to thank our head of the department, R.A. Manjunath, for taking a step towards our special lecture program and make it a grand success. Once again, thank you, sir. I
Thank
would
you.
also like to thank our President and Honorable Secretary for their constant encouragement and support. I thank you, sir. I would like to thank Dr. Sri Raghavan and Dr. Aisha from the Department of Biotechnology and Mrs. R.S. Preeti from the Department of Microbiology for their support. Thank you, sir, and thank you, ma'am. I would like to thank all the and behind the screen who guided through this session. Thank you. I would also like to thank all the technical staffs, teaching and non-teaching staffs, and the students who are participated in this session. Once again, thank you one and all. Have a great day.
Thank you, Tejaswini, and thank you one and all for joining us with and participating